with what's going on in the markets is, I think, a widening gap between early stage drug development and a point of partnership or investable assets that would help some of these therapies advance to patients. And so we all have an obligation to be really creative and understand what are some of the new paths that drug developers could take. And in rare disease, oftentimes those drug developers are advocates, they're parents, they're citizen scientists. So how can we help guide them to move forward so that we have a continued productive pipeline of therapies that are out there, in particular for those ultra-rare conditions that might look to a lot of folks as not being commercially viable.
Great. So I'm Rodney Samaco, and I will be serving as the gentle guide and moderator for today's session. And I will let themselves introduce each other. But first, I would like to say, to echo what Craig had mentioned, we really wanted to think outside of the box and do something different. So COVID has really disrupted, as we all know, in the past 2 years, a lot of our lives and a lot of the efforts that we have built in infrastructure and capacity. And so coming back together, we brainstormed, and this is not just me. Behind me, or with me, I would say, are also the coordinators from Orphan Disease Center and Global Genes, and thought, well, what can we do to really make this appealing and really make this meaningful in terms of gathering again? And so we considered, make it interactive. Let's change the format and instead make this completely disruptive. And so the panelists today will be discussing some of those key factors that could accelerate research and make the entire process end-to-end disruptive at scale, and discuss the challenges and solutions, positive exemplars of how they could really get a tangible hold on making something great. Okay, so go ahead and introduce yourselves, and then we'll go from there.
Okay, great. I'll start. Good morning, everyone. Thanks for having me. I'm Carla Rodriguez-Watson. I'm an epidemiologist by training and the director of research at the Reagan-Udall Foundation for the FDA. So the FDA's foundation is to help the FDA achieve its mission to enhance product safety, innovate on product development, and to spur innovation through regulatory science and primarily through private-public partnerships.
Good morning. My name is Paul Howard. I'm a political scientist by training. I am currently Senior Director of Public Policy at Amicus Therapeutics, where I think a lot about how to scale the work that we do across our many communities, whether it be our patient advocacy partners, the research community, our regulatory partners, so that we can break down silos, bring data, bring information, and even learnings from other industries together to help us accelerate the challenge of developing better treatments and hopefully potentially cures for rare disease patients.
I'm David Fajgenbaum. I'm an assistant professor here at the University of Pennsylvania, where I run a center called the Center for Cytokine Storm Treatment and Laboratory, also an associate director of Penn's Orphan Disease Center, and I run a foundation called the Castleman Disease Collaborative Network. Today, I'll try to contribute a bit, both the physician-scientist perspective as a researcher in rare disease, but also as a patient. I was diagnosed with a rare disease called Castleman disease while I was a medical student, and have gone on a journey that so many of you all have to start foundations and to try to drive forward the science, and hopefully those perspectives are helpful.
Thank you. So we had a very robust discussion. Maybe you could pull your chair a little bit, and I'll pull my chair this way, as there's a fire in front of us, as, representing the Fireside chat. We had a very robust discussion when we first met in preparation for how can we make this meaningful and engaging for the attendees. And we talked about some of the challenges in this space. I'm going to start with a primer, which is what are the tools and standards we could create working together as a community to help us better understand phenotype, genotype, and get, as you all said, better shots on goal.
Anybody?
Anyone?
Anyone?
You were very talkative, all three of you. So
I'll just throw some ideas out, and humbled by sitting next to Carla from the Reagan-Udall Foundation and David and everything he's doing for Castleman's. But I think a lot of what we're talking about is how to improve the quality of data that we're collecting, how to be more responsible with the patient data that we're collecting, more transparent about what we do with study participants' data, and ensure that we're not reinventing the wheel. I think, in one area where we look at something that we've talked about for a long time, which is, a synthetic control arm. We industry, have lots of trial data. How can we come together to create high-quality synthetic control arms so that maybe when we run a trial, we don't have to have a control arm. We can have an active arm and a synthetic control arm, and that can reduce the need for patients to be exposed to a placebo and potentially get answers faster. So I think that that's one of the areas to start thinking about. And there are data collaboratives out there that are emerging. I'm sure Carla knows more of them than I do, but one of them I know from another field is Project DataSphere. It's in oncology. I haven't looked at their website recently, but when I was at the FDA a few years ago, we worked with them quite a bit. The Oncology Center for Excellence worked with them quite a bit. And they brought together, I think, 8 to 12 large pharma companies who contributed control arm data from lung cancer, breast cancer, prostate cancer, and they routinely published data from those data sets, and even had X Prize kind of competitions to see if they could develop algorithms that could predict, say, based on control arm data, patients who would be at high risk of progression for prostate cancer. So I think that that's one of the potentially low-hanging fruit that we could start thinking about as a community, so that we could be better stewards and be more responsible with the data we've already collected.
Yeah, I totally agree. A couple things I would add is the really foundational elements for us for Castleman disease to make progress were that we needed to first be able to connect with our patient community to prioritize what research should be done. So we created a tool called CDCN Connect, where patients can actually contribute what are the research questions that are important to them. Physicians and scientists can contribute what research questions could be important to patients. And really, we use the term crowdsource, but it's really just get a sense for what's important to patients. That's a foundational step one. Another couple of foundational steps are building a biobank and a registry. And so if you can tie together really high quality biospecimens from patients along with medical record data, then all of a sudden those biospecimens become really, really useful for doing science. And if you then do the science that's important to patients, you end up making a lot of progress, in our case, in a short period of time. And I guess the last thing that I would mention in terms of tools, is particularly related to data, is around drug repurposing. So I share a lot, and I'm a really passionate advocate for the concept of drug repurposing. So that's taking a drug that's already FDA approved for something and figuring out a potential new use for it. Obviously, in you all's context and in mine, for my rare disease, for your rare disease. But it's really important when drugs are repurposed or they are used off-label, it's essential that you collect that data. What you don't want is to try a drug in a patient, have it work or not work, and not learn from it, because then the next patient, we can't be more informed when we go to treat them. So registry, biobank, a solution for crowdsourcing, and then make sure you track the data if you do any drug repurposing.
Yeah. I think what you've both talked about is really talking about how you bring different stakeholders to the table and identify what are the issues in a pre-competitive space that you all need to attack to address challenges and find opportunities. One of the things that you both mentioned, I think, David, in developing the Castleman disease registry, is the opportunity that gave to bring together the industry, as well as the patients, as well as the researchers, to start to think about how you would further describe the disease. And you said from identifying 2 subtypes to 6 subtypes, and really classifying what Castleman's looks like. And we did something similar like that, and Rodney, you mentioned COVID-19. It did stall a lot of different work, but it also spurred a lot of work. It also spurred this idea of collaboration and coming together and being able to share. So one of the things we did in the convening space was to bring together leadership across the healthcare ecosystem in government to crowdsource solutions, data solutions, and ideas about how to address prioritized research questions, this time not necessarily from patients, but from the FDA, because there were specific data problems that they wanted to address and to understand better. And so we were able to bring together people in a way that was pre-competitive and helped kind of this idea of rising tides lift all boats. So I think focusing on those areas that would benefit all, that don't conflict with or impinge upon intellectual property is a key. And there are, I'm sure you could think about many of those solutions out there.
So what comes to mind is a few questions that I have that maybe you could help us better understand how these actually came to be. So when you talk about data silos and the pre-competitive space for people to share data, what were the challenges there, and how did you overcome what I like to say, there's no technical solution to human behavior? A lot of the unpredictable nature of how different stakeholder groups may interact with each other, and trying to understand those incentives and right aligning them. How did you make that happen?
I'm happy to jump in around Castleman's disease, and would love to hear you guys' thoughts as well. But Castleman's is a rare disease, about 5,000 patients diagnosed each year in the U.S. And so as a result, there are centers around the U.S. and around the world that had patients and also had medical data and certainly samples as well, but there was no process for physicians and researchers to share that data. And so we started out really early on with the CDCN trying to figure out paths for data sharing and sample sharing. And we spent a few years really trying to do that, and we really struggled, frankly. It was hard to get agreements between academic institutions. We couldn't persuade a lot of researchers to share their samples, in part because those samples kind of served as assets for writing grants and for being the leading experts in the field. And so at a certain point, we still try to facilitate that, but at a certain point we realized, well, maybe we should maybe just go directly to patients. And so that's where we created a registry and a biobank where patients consent directly to our organization. And then when they consent to give us the samples, we get a blood sample from that patient, we send them to a local Quest Diagnostics. We sort of eliminate the medical system and the physician and the researchers from that process of getting samples, and then we make the samples freely available to scientists. And so what that's done is it's meant that we can get the samples and the data directly from patients, and by then giving those samples and data back to researchers, that's made the community a bit more collaborative. So those people who are maybe holding on to samples and data are a little bit more willing to share because we're sharing with them.
David, can I just follow up on that and say, is one of those advantages, there are a lot of incentives that push us into different silos. But from the perspective of regulations now that encourage patients to be more data empowered, to own their own data, even if one researcher says, well, I collected this data, that doesn't prevent the patient from taking their records and sending them somewhere else, or giving you specimens. And I think one really interesting component of this, and Craig Lipset and I were talking about this on the sidelines last night. Patients, I think we have an obligation to tell patients what we're doing with the data, report out routinely on the data uses and data quality, and just be more transparent about that. But I think also, as you did, I think it's critical, go right to the patients themselves and empower the patients with the information they need to make progress. And at the same time, hold the rest of us accountable and ask us, what are you doing with my data? How are you sharing my data? So I think that kind of patient-powered, demand for transparency and accountability, for like what happened to the cells of Henrietta Lack? What's the story there? Why did that happen? Let's not let that happen again today. As we're asking more and more questions and want to do more and more things with the patient data and specimens we collect, we have more and more of an obligation, I think, to be transparent and ethical in how we use that data.
And patients want to contribute their data, right? They do want to engage. As you all know, you want to engage in this research, but there is a need to be responsible. And there's lots of more and more organizations. I was just talking to Caitlin at Cross at All Stripes last night with Inga of the Autoimmune Registry. They're going out and reaching patients and creating these registries that have patient-generated data, including survey information, that can augment and provide outreach for trials and studies, and enable them to opt into certain studies, as well as potentially linking to their clinical record. So that could be very powerful. And there's definitely, the FDA has issued guidance on more and more inclusion of patient-reported and patient-centric study designs and is doing more work on that. The CTTI has an initiative for 2030 on its goals for clinical study designs, which include increased patient centricity. So there's definitely a focus, and we were talking in a group last night. I think although there is guidance, there is a need to operationalize that guidance. And so having folks in the researchers in the community and sponsors that have stuck their neck out to do this work, to come forward and say, this is what we tried, it worked, and this is where we had challenges. And then work through that, and having regulators, not just the FDA, but EMA, others, at the same table to hear what the challenges are and start to work towards solution.
When I think about challenges in terms of communicating, right, and healthy relationships require iterative processes of communication in all spheres of life, we'll say, right? Some of these aspects that you're describing are really, I would say, come down to communicating well and articulating exactly and explicitly and being transparent with what the end goal is. So how have you had those conversations to work? So I'm really trying to dig deep here in terms of examples that you could provide in which there were challenging or hard questions to really bring up to the room, to the table, and you had to manage the conversation, manage that difficult conversation to find solutions towards a common goal.
Yeah, I think one of the most difficult conversations we had at Amicus, and this actually happened before I came on board, I guess in 2018, we had a program for epidermolysis bullosa, and we had interim positive data in a phase 2B trial, and ran a, I guess, 3.5 year long double-arm trial. One arm was randomized, controlled, double-blinded, and another one was an open-label trial. And after they unblinded the arms, they found out there was no difference between the treatment and the placebo. And that's devastating information to have to convey to patient communities and patient partners. This is a extraordinarily difficult and challenging disease that has enormous burden on patients and family caregivers. 30% or more of the patient body area of EB patients can be covered with lesions and wounds. And so anything that even makes a small difference in those patients' experience of pain and comfort is extraordinarily important, and we thought we had something, and it turned out that we didn't. And our response to that was to convey as much information as we could about what we did and what we learned as possible, in the hopes that regulators, other companies, the patient community themselves could learn from the trial. There's no such thing as a failed trial. You learned something. You collected some data. You now have a better understanding of the natural history of the disease if you've done the study well. And we published peer-reviewed papers. We presented posters, I think at a Global Genes conference. And we met with all of our patient partners to explain to them exactly what we learned, exactly why we were closing the program down over what period. And met with, I think, 5 or 6 companies that were also working in the EB field and gave them our data and our research. One of them, Amarit, took it forward, and I think they recently concluded a phase 3 trial. So I think that one of the things we have to do, again, with that theme of being incredibly responsible with study participant data is finding a way to communicate as much as you can, as early as you can, while still protecting patient privacy and complying with regulatory authorities, but especially when a trial concludes, and most trials are going to conclude with a finding that you didn't want. You were always hoping for a positive finding, but if you have a negative finding, you made it somewhere down the field, and you can help the next person to pick up that knowledge and go even farther. So I think that's one of the things we can do.
I just want to follow up to that to say that that's just amazing. I mean, I love hearing that, and I think that too many of us have heard stories that haven't been so positive about data sharing. I mean, that's just incredible that they went out, they found other companies, they shared that information. It's not always the case. It should be the case. I love hearing that. I'll also share another somewhat related example where there's one FDA-approved drug for Castleman disease. It's called siltuximab. It works in about 1/3 of patients. And I had an important conversation with the makers of siltuximab at Janssen Pharmaceuticals, or Johnson & Johnson. And one of these employees at Janssen informed me that as part of the clinical trial that led to siltuximab's approval, they also collected tons and tons of blood samples and lymph node samples from those patients that were part of the trial. And those samples were sitting in freezers. And in fact, the patients consented that the samples could be used for a number of purposes, but it had to be within 10 years of the trial concluding. And we were about 8 years into that 10-year window. And this colleague at Janssen didn't have to tell me about this, and he knows who I am, and he knows that if you tell me that, I'm gonna basically break down the walls of Janssen to get my hands on those samples. And so, he could have, not shared that, but he did. And once I learned that there were these samples, it started what ended up being about a 2-year process. We got it done just in time, but a 2-year process to work with colleagues at Janssen, also the Castleman Disease Collaborative Network, and the University of Pennsylvania, and then actually, I think 7 other institutions around the world, to put together what we thought was the right kind of study to leverage these samples. Patients had already consented for their use. They were just literally sitting in freezers, and we ended up doing a serum proteomic study. We used a platform called SomaLogic, where we measured 1,300 proteins in the blood of all of these patients. And we learned so much. We discovered a biomarker so we can predict who's likely to fall into that 1/3 of patients who's going to benefit from siltuximab, who's not likely to respond, and maybe most importantly, we identified a novel therapeutic target that we can go after in people who don't respond to siltuximab, JAK1/2. And we've actually treated patients with this JAK inhibitor. Within a couple weeks of the results, we treated a patient in Chicago who was really struggling, and she's been doing well for almost 2 years. That company, Janssen, didn't have to go through the hassles and the headaches of working, with me and my team to put these contracts in place, to get these samples exposed, but they did, and it's literally saving lives. Like today, patients are on drugs as a result of it, and those drugs are not made by Janssen. Janssen is not making any sort of, there's nothing positive for them. It was just a lot of headaches, dealing with me and with the attorneys. But it's been great for patients. And so, I just think about all of the trials, like Paul mentioned, where clinical trials are done, and just as you said, no trial is a failed trial. At the very least, there's data, and sometimes there are samples. But oftentimes, those samples sit in freezers for years and years. So what can we do as advocates and also members of the pharmaceutical industry to work together to unlock those samples from freezers and to really learn from each of these patients? And the last thing I'll say is that a number of patients that were part of that siltuximab trial are no longer alive. The final remaining opportunity that those patients with this disease had to make a difference for future patients were sitting in those freezers. And those samples from those patients who are no longer here are helping patients who are alive today. And that is a concept that I think is just so important, and something that, whether you're in the pharmaceutical industry, academia, advocacy, we should work to unlock those samples.
But it strikes me that there has to be an incentive, right? Like there was some incentive for you, Amicus, to share that data. I mean, whether it was like shared publication going forward, or to take it forward, and for Janssen, what could we call something out that we could have as a carrot?
It's a good question. I mean, I think that Janssen, maybe they
Or that everybody
made
knows
a
that
mistake
Janssen's a superstar, and everybody should take the J&J drug now.
No, I don't know. I mean, what would you think? What would you say is the motive? Is it that you and your colleagues really care about patients, and so you felt it was the right thing to do? Because I do think that really was a big part.
I think a lot of it is because the CEO of our company, John Crowley, has 2 children with Pompe disease. He's a rare disease parent. Mm-hmm. And he knows the pain these families go through and the hope that they have when they go into a trial about what the outcome is going to be. And we know that that's still extraordinarily challenging. But that's part of our, if you'll pardon the expression, part of our DNA and part of our culture is to be as patient, participant-centric as possible. And I think that a lot of the silos that are out of there are lack of incentives and kind of clarity about the why. And the why is that we really can advance science faster if we do it in common and hold less information. There are a lot of incentives that encourage us to be less forthcoming with our data around intellectual property and patents. But there's still lots of opportunities that David could find a biomarker that turns out to be a potential surrogate endpoint for a new drug.
Yep.
Right.
That's right.
And so that benefits everybody.
It benefits everyone.
So in the power of those numbers, I think we've approached this also, and I've heard other in the registry world talking about this also, when you're looking at rare diseases that have some underlying common basic pathophysiology, like the autoimmune registry, right? There's any number of many, many autoimmune diseases, and to try to ramp up a trial for just one would be hugely challenging. But maybe if you can group them for those that are more gastro versus dermatology, right? Is there a more different study design approach, like basket trials, that you could conduct? And then so you will be able to increase your sample size in that way, and explore what are more common surrogate markers among them, and look at, it could be more efficient for drug development and understanding drug safety to look at one exposure for many different indications. And I know that the FDA has issued guidance on master protocols. That is being pushed out there more and more, and people considering these trials in oncology. We're thinking about them in terms of pregnancy. Pregnancy is a rare disease since you have all this labeling that says don't use this drug when you're pregnant. So, finding those are, especially in real world data, becomes more advantageous because if it's being used, you can find its use off label as opposed to in a prospective registry where people have to actually sign up and abide by specific, very tight inclusion-exclusion criteria that might knock them out. So, commonality.
We still have plenty of time. Yeah. So maybe you could elaborate on that, as that was one of the solutions, the takeaway messages from our preparation discussion, using pregnancy as an example. Do you want to elaborate on that?
More. Okay. I think I've said it. I mean, pregnancy is like a rare disease, and so we are thinking about conducting basket trials using master protocols to look at exposure for one drug across multiple conditions that they're autoimmune. So looking at those that are more IBD focused to be more efficient and increasing the sample size for one of the drugs.
So, like David, you've said this, not to throw the ball at you next, but you've said that there are a lot of commonalities between cancer and Castleman's and some autoimmune diseases. So when you compare the gene expression maps of patients with those diseases to Castleman's, what are the commonalities? How do you think about exploring that shared space that revolves around the human immune system? And what are the kind of shared learnings that maybe we can think about borrowing and learning from faster, looking at what people are doing in oncology and what they're doing in autoimmune to learn?
I'm really glad you asked that, because I think this is an important paradigm for all of us within the rare disease space to consider, and that's that every rare disease, there's typically a cousin disease that's more common, that is related in some way. And oftentimes, when someone would ask me the exact question Paul just asked, in Castleman disease, I would say, well, it's a good question. There's probably similarities, but we've never done the profiling to really compare them. But with that proteomic study that I mentioned, that we got those samples from Janssen, we ended up also pooling together 20 patients with rheumatoid arthritis, 20 patients with HIV, 20 patients with Hodgkin lymphoma. So we had an inflammatory or an autoimmune disease, a cancer, and also an infectious disease. And we could then ask the exact question, what is Castleman's most similar to? And what we found is that it really, it's broad spectrum and it's very heterogeneous, but we found that the patients that had proteomic profiles most similar to the Hodgkin lymphoma patients responded best to an IL-6 blocker. The patients that had a proteomic profile most similar to the rheumatoid arthritis patients seemed like they were the most likely to benefit from a JAK inhibitor. JAK inhibitors were not made for Castleman disease, but as a result of that profiling and the profiling we did in the what are called enrichment analyses we started treating patients with JAK inhibitors based on that information. And so I think that so often we focus on a rare disease, and I'm very guilty of that, but I think that we need to take the blinders off a little bit and say, when we do a study, whether it's EB or Castleman's or whatever that study may be, let's ask what are the cousin diseases that are similar, especially if they're common, so that we can really learn from one another and really make sure you include those samples when you do studies. It's a great question.
Yeah, I mean, I think Mother Nature is lazy. And once she finds a pathway that works, she tends to keep using it. And one of our challenges and I think my colleagues and I talk about this all the time, when we think about rare diseases like rare CNS diseases that might have common symptoms that affect core clinical features of the disease like gait or sleep or speech or tremors and seizures, there are a lot of diseases that fall into that bucket. And I know there are efforts underway, for instance, to try and take what we think of as a large disease and disaggregate it into different etiologies, like Parkinson's. Is that one disease? Alzheimer's, dementia, is that one disease? It's probably multiple diseases. But we can use a set of tools, wearables, sensors, algorithms that can help us understand the phenotype back to the genotype. And there I think there's an opportunity to recognize that you do have those shared symptoms. There may be opportunities to use a digital biomarker or a wearable to develop a better understanding of the natural history of the disease, and also see if you can modify that pathway within a targeted approach, like bringing the genome and the phenotypic data together in a really fine-grained way with a tool that can give you 24/7 data on patient feeling and function that can help us kind of tease out those profiles and look for commonalities.
Yeah, Carla, I don't know if you have anything to add to that.
No, I think that you've all really tapped into it. I don't have anything else to add.
Yeah,
Do you think there's,
sorry, I was
Yeah,
gonna-
no.
I was
Do you
gonna-
think there's any disadvantage in substratifying or stratifying?
Well, I think that when we're talking about this kind of aggregation and looking at it, like in the example of approaching it from a basket trial approach, and in rare disease, you could look at the totality of it and look at those that are exposed versus not exposed to the drug and see if there's any great difference. I think in stratifying, the issues come with the numbers, and then that's where you could get hung up. But a way to expand beyond that, especially if it's about contextualizing or understanding the natural history of the disease and the epidemiology of the disease, is to look at real-world data. I think, as you've mentioned, you looked at HIV patients, oncology patients. There are federated data systems that include these large cohorts of different populations in HIV, in oncology, and autoimmune, and other diseases. And so that's an opportunity, I think, to identify patients for trials, to do pragmatic trials, like you were saying, like in the clinic, so that they don't have to come to a center and take all that burden of the time it takes to come with their one child with a disease, for example, if it's a parent, and what are they going to do with their other children who are left at home because of COVID restrictions, and decrease that burden and engage them in decentralized, pragmatic trials. Leveraging real-world data and being able to identify those patients more efficiently would help address some of those challenges that you get with those small cell numbers.
I don't see too many downsides to really subsetting or subphenotyping rare diseases. I think that the smaller the unit, the easier it is to understand what's happening in that patient. And to get back to one of Paul's comments around shared biology across diseases. We all have the same 20,000 genes, and with at least within certain diseases like in the immune system, there are dozens of pathways that are shared between diseases, and that's it, just dozens, right? And so as a result, that's again where I would get back to this concept of drug repurposing, is that a drug, whether it was made for rheumatoid arthritis or for Castleman's or for COVID, you're targeting a particular protein or pathway that often is shared. And the couple of examples that I'd highlight, one is the drug tocilizumab that was initially made for Castleman's about 30 years ago. It's the first drug that you get if you're admitted to the ICU with COVID-19, and it's probably saved hundreds of thousands of lives right now. That was a rare disease drug made for Castleman's. Now, obviously, in that application, going in the other direction, the drug that I'm on that's saving my life is a drug called sirolimus that was initially developed for kidney transplant rejection, to prevent rejection of organs, and it targets a pathway called mTOR, and we did the work to figure out that mTOR was important in Castleman disease and said, well, that's great. There's a drug that inhibits mTOR. I don't care why it was made or how it was discovered. It targets mTOR. Let's try it. And I think asking those sorts of questions, I mean, it saved my life, and it saved a lot of other people's lives. But we have to ask these questions of what is the shared biology, and you have to have the data to really be able to make those inferences.
There's a word that you said at least twice, and I say you, I think Paul and David, you've said it, you may have said it, Carla, and it's the F word, which is fail. And I hear that often, with clinical trials, and you see that published, in various media pieces, et cetera, that there's this failed clinical trial, et cetera, fine. But here I'm hearing that it's not a failure, that we should take this as a teachable moment, as lessons learned. So why do you think that we continue to use this negative word, failure, that has negative connotations, and instead, why not call it as it is, as a misstep? And you can answer that in any way you'd like.
So, it's a learning, right? It's a learning. I think, but why do we call it, or how can it? I can give you an example in addition to what Paul said of how we took a learning opportunity to further a cause.
Learning, teaching moment.
Learnable moment, teachable moment. So in COVID-19, in our COVID-19 Evidence Accelerator that I've described before, which was really the Reagan-Udall Foundation for the FDA in collaboration with Friends of Cancer Research started this at the start of the pandemic to bring together leadership, not just to have a forum to share information, but also to execute on prioritized research questions from the FDA, where we crowdsourced from the community there that we built, which was over 250 organizations that came together and still come together in monthly meetings in a diagnostic space and in a therapeutic space to talk about COVID-19. In the early days, it was about understanding epidemiology, cure, treatment, and now it has kind of morphed into understanding lack of data interoperability. So one of the questions we were focused on in the diagnostic space was trying to understand the real world performance of these emergency use authorization tests. And to do that, you need to be able to link the manufacturer data as well as the results, the clinical presentation of the patient, all those things. And what we quickly found out is that unlike my iPhone or the coffee grinds in this cup, there is no UPC code for a diagnostic, for a test. So there is a device ID, it's out there, it's out there, but it's not transmitted. And so one of the important things, we were not able to answer that question fully because of 6 partners that we engaged on that study, half of them had more than 30% of the manufacturer ID missing. And for the other half, it was a patchwork of I know somebody, I know a guy who knows the ID, the device that was used. And that's within one system, right? And so when you're looking at data aggregators that are dealing with multiple network systems across the country or insurance claims data that don't get that information, and that's a lot of the engine behind real world data. When you don't have that missing, you're not able to answer the question. And so there was a real opportunity to, I think, understand the most important outcomes when the agency was pushing out these emergency use authorizations, and there was this hope that we could look to the real world data to help us get more informed and understand its effectiveness in real world practice. But without having that piece of information, we weren't able to completely describe the problem. Where we did, and where we were sure we were transparent about what was missing, and where we could be confident in the inferences, we reported that. But that work and that gap spurred the agency to further it and to solve and put money behind solving that problem, so that this year, the FDA's foundation issued an RFA, and we funded $2 million to go to 2 organizations to produce a framework to identify and try to assess the real-world performance of over-the-counter diagnostic tests and PCR tests. So to do it with real-world data and figure out a way to move that information across.
Was the problem that you couldn't link that individual EUA test back to an individual patient to compare it to to a PCR test? Like, was that where it broke down?
Yeah, I mean, to link the actual test, the manufacturer test, whether it was Abbott or Roche, for the PCR and for the serology test. We were looking
the
at
rapid test.
Yeah. So, well, we were looking at serology. So in our example in Evidence Accelerator, we're looking at the real-world performance, the positive % agreement of the serology for positive PCR. So in PCR patients, just to see if we could link the data and do that assessment. But we found the gap in linking the tests that were performed to the patients themselves and the result. We knew what test type, but we didn't know the manufacturer. So that's kind of where we took it a step forward, and with the support and the interest of the agency, we're moving towards answering that question. So like you weren't able to answer that question, you said in Amicus, and then you moved it forward to another group that could, with other resources, additional resources, whatever, is furthering to address the problem.
And I love the concept of thinking about these failures as being learning, teachable moments. I think it's in line with the idea of we shouldn't just look for silver linings, we should look to create silver linings in the midst of really tough times. That said, a trial that doesn't achieve its primary endpoint is devastating for patients and for families. It's a learning, teachable, all this stuff for scientists and for companies and for all this stuff, but it is a heart-wrenching, devastating moment. My mom had cancer when I was much younger, and when we learned that the trial that she was on was not effective, that crushed us with everything. So it was a failure. I mean, obviously, I'm sure at some point there were some learnings from that, but from a family member and a patient perspective, a trial that doesn't meet a p-value of 0.05 is devastating. It means that your loved one's not going to be able to get this drug that you thought was going to be really hopeful. So, of course, we have to create a silver lining in that. It is devastating. Let's create a silver lining as a healthcare system, as an R&D engine. We have to learn from it. But I think that the reason we use the word failure is because of how horrible it is when these drugs don't have.
Yeah.
It's how it feels, right? And it's no one's fault. But we all tried the best we could, but it's horrible.
And I think that, again, there are incentives. So, there are incentives for researchers, academic researchers who want to continue to publish on their data, and often for quite some time after that study or effort might conclude. There are companies that have that data and think, well, this might not have worked here, but could give us an advantage on our next project. Or if we put it out in the public domain, could give a competitor advantage on their next project. But maybe there are tools and opportunities for trusted stakeholders like Reagan-Udall to use federated learning techniques, like you had mentioned, for companies to be able to, query data that might be held by the agency, might be held by independent companies around specific questions that we might have around the data, that we could come together and develop a short list of, core clinical and genomic features we wanted to answer for a given disease state, and then begin to try and build them out in a trusted, neutral convener environment where we could all learn from the data together at the same time.
Yeah, absolutely. And I think that's really a space where the Reagan-Udall Foundation is able to support, is bringing different stakeholders together to share that information and making it feel like there's some neutrality to it. But I think to some of these, I'm just curious, when you say the trial failed, is that because the sample wasn't big enough? Or many other things? Was it powered? And then, I think, again, using real-world data to identify more people.
I think it was if if I recall correctly, at the time it was the largest trial run in EB. I think it was 169 patients. It was a 3.5-year-long trial. So I don't know. I mean, the problem with trials in rare disease patients is you don't know enough of the natural history of the disease to say that, well, there's an improvement, or is this just a change in the natural course of the disease that might look like at a certain point in time there's improvement, but later on turns out to be not. And then when you compare it to a placebo, you say, oh, that wasn't what we thought it was. I mean, I think that one of the biggest challenges that I think industry faces is by the time we take a real shot on goal in a phase 3 trial or a late phase 2 trial, we've already invested a lot of time, and we need to be able to take much smarter bets, make much smarter bets earlier, to take, an iPSC cell line data that might have been produced by an algorithm looking at thousands of gene expression data points and existing trials, and take it as quickly as we can into a lab bench test. We need to run as many experiments as we can in silico first, and then go to a plausible biological construct, an animal, maybe a cell line, and just see if we get a hit. And even a small improvement in that 10% would be, earth-shaking for us. So I think that a lot, we're all in a search for better data, I think, at the end of the day, that can help us make smarter decisions about what products to take into the clinic, develop better animal models, and especially use some of these new approaches to look for biomarkers or panels of markers using machine learning that we might not see, that a human being might not see, just because we can't analyze that much data. I think those are the opportunities, and we want to be able to fail much more cheaply and quickly.
Would being able to identify patients using AI from real world data be able to increase the sample size that you might be able to leverage? And would that help to better characterize and then improve the downstream pathway?
Yeah, I think it could. I think there are a lot of people who are looking at this. I think HealX is a company that's looking at using a combination of genomic and facial recognition data to try and shorten the diagnostic journey. And if you can use AI to, for instance, because we know that a lot of patients will go 7, 8, 9, 10 years without a definitive diagnosis. And that can change everything. If it changes the number, the incidence of the disease, that could draw investment. If it helps you disaggregate that population into more tractable disease states, I think that could be incredibly powerful. So yeah, absolutely.
Yeah.
I think real world data could be a game changer.
Yeah, and also because a lot of these conditions have endpoints that might not, the traditional endpoints might not be those that are important to patients, right? And then some of these autoimmune conditions like brain fog and chronic fatigue are not easily recognized through laboratory. Sometimes your labs stay stable, but if you have patient symptom trackers that are able to, and we can do better at characterizing and standardizing that description in some way of a patient's symptoms that are not recognizable from traditional like laboratory values or diagnostic tests that are run. Would that first improve the patient centricity, the outcomes that are important to patients, as well as be able to, like you say, be able to classify or categorize disease and increase your sample size potentially?
Absolutely. Yeah. Well,
because most of those people could be underdiagnosed,
yeah.
right?
You get a lot more data points. So yeah, I think it could make it higher powered.
Yeah, the only other thing I would add to this Carla's great question about, what do we mean when we say failed? And I think that it is important to break down that into a few different categories. So one is that the drug just probably doesn't work, right? It's not any better than placebo at improving the thing that you want. Hopefully it's something that's patient-centric. Second is that it looks like it works, but it causes horrible side effects, just too much that we just can't move this forward. And third is that maybe it is a study design issue where the study wasn't powered. You didn't have enough patients in the study. You didn't pick the right subtype. You didn't have the correct inclusion-exclusion criteria. And I think the third one is the one that maybe is the easiest for maybe a patient group to swallow because, okay, well, maybe there's still a chance that this is going to work for my disease, but it's probably the hardest for industry to handle because it's, oh my gosh, if we just enrolled more patients or if we just thought about the design differently. But yeah, I think that it is important when we say a failed trial, not all failed trials are equal. It could be, any one of those things.
Mm-hmm. So we have about 5 more minutes, and in the last 5 minutes, maybe you could touch upon each of you. So time is not a resource that is replenishable, right? It's a limited quantity. And in many of the clinical trials that you described, you say it may take 2 to 3 years, or you set up by repository 2 years, et cetera. And I would say many of the attendees here, one of the reasons why they're here is because they don't want to wait. And I'm sure we all agree. So what would you suggest, if you were to suggest one solution to move the path much more faster, to accelerate their rare journey? You could think about it. I'll give you 1 minute, unless you have an answer now.
I say find your friends. Understand how your rare disease has a cousin with a greater population prevalence. Try to find as many of you to get together. Identify diverse stakeholders of industry, patients, researchers, data aggregators, healthcare delivery systems that might provide data, all kinds of data sources, as well as, did I say regulators? And get together and understand what's the pre-competitive space? What are there biomarkers you need to understand? Do you need to better describe the disease? Better identify important outcomes that aren't traditionally collected? And get to work, develop those outcomes, and work on the pre-competitive stuff so that that rising tide lifts all boats.
Yeah, I completely agree with everything Carla just said. I mean, I think that there are a couple things we need to do, and I don't have any good answers. One of which is I think we need to think of better business models. I mean, we need to align the incentives that encourage data sharing and high quality data generation. And because, I read David's paper, and the amount of data curation that went into it is just enormously time consuming. And I was struck by the fact that patients donated more data than the sites did, the physician sites did, because the sites didn't have the funding to go back and get all of this retrospective data and curate it. And I think, what kind of tools can we create, what kind of new business models can we create that we can make it easier for everyone to access and curate highly tractable, valuable data that makes potentially a lot more sites, clinical trial sites, that they can enroll patients. So there's an opportunity there for HCPs to earn additional income, opportunities there for patients to enroll in trials and participate in trials and where they live and work and not have to travel 6 hours or give up time with their families or lose money from their jobs, because we can use decentralized trials and pragmatic trials to help treat them where they live and work. I think we need more business models there. And then I think for us as a community, we need to think really hard about where our value added is. Just like when someone starts a new business line and what's going on. I mean, competition in the sense of where are people really adding value and where is no one looking? And try and figure out that space where no one's looking or no one's doing anything that really could help. Don't go to where everyone else is working. Go to where no one is working, but someone needs to be working.
Totally agree with both these suggestions, and I'm struggling to decide what is the one thing. But
You
I
can
think
say more than one.
okay, thank you. I think that I would really dig into this idea of unlocking samples. If there's ever been a clinical trial done for your rare disease, or if you have a biobank of samples, or if you can go directly to patients to get samples, just because a rare disease has a gene that's understood to be a driver of the disease or seems to be well characterized according to an animal model, I'm a huge proponent of profiling human biology. Let's obviously animal models and cell lines have their roles, but the patients are the ones that have the disease, and what's happening in their bodies I think is really critical. So unlock samples, use unbiased profiling techniques, whether it's proteomics or transcriptomics or a variety of other approaches, but really try to understand what's happening molecularly, and then always ask the question, what drugs are already FDA approved that are known to potentially be able to abrogate or to modify the pathway or cell type or signaling path or gene that may appear to be important based on your profiling. What's happening, and then what already exists. There's 3,000 FDA-approved products that hit many different targets throughout the human proteome and genome, and just because they were made for one thing doesn't mean that's the only thing that they're destined for. And so profile and then think about repurposing.
And use real-world data to find the people that are on those already approved drugs.
Yes, exactly.
And get them into your trials.
So thank you so much for sharing your insight. We can't take questions, if there are any, in the last 10 minutes. We had set aside a little bit of time, unless you want to break out and address them within the sessions. Because you're all three gonna be here both days, right? Okay.
I did see a hand.
Oh, you did see? I can't see because the light is. You could walk up to the microphone and.
Thank you. Okay, that was, you gave me so much to think about, and I really appreciate it. Laura Pullen from Xi'an Foundation. Our focus has been on Prader-Willi syndrome, and one of the things I want to go back to is something you said early on. The registries, I think, are incredible, like this great opportunity. But so I'm most familiar with Prader-Willi syndrome, PWS, and our registry is kind of locked down. So we had an outside hypothesis, and we couldn't access the data in the registry to explore the hypothesis. And it seems like that's standard for these registries, that whomever's running them sort of owns it. And I get there's this tension and there's balance for who owns the data. But it seems to me like the rare disease world would be a better place if the David and the Rodneys of the world are working late at night and they come across PWS and they want to dive into the registry and look at the de-identified data and like sort of poke around and say, so wait, is sleep a thing in PWS as well? Or what do we know about the mTOR path and, or whatever? Like, it seems that, and I guess I wonder, when I poked at the PWS organizations as to why it wasn't available, they just said, well, you know, people can't be trusted, or it's not standard, or there was no really good answer. And I wonder what you all think about it, and if there's something that can be done and said by Global Genes or here at this thing that, like, to sort of endorse this opening up of that data to help break down silos and to find your cousins and explore, like, out-of-the-box hypotheses. So that's my question.
It's a great question. It's something that Paul and I have spoken about before. This is a huge problem. You know, there are many registries. In fact, for some diseases, there's multiple registries just for that rare disease. And the data is not shared. And in a lot of cases, there are mechanisms to access the data, but whoever owns it sort of kind of pretends or makes it seem like it's impossible. And I think in a lot of cases, the people who are doing that actually have good intentions. They oftentimes, I think that they're the only ones who can interpret the data because, like, they built it, they know the patients. So, like, everyone else would be confused if they looked at it as kind of their mentality. I totally disagree with it, but I don't think it's like they're thinking, like, I want PWS patients to suffer because I don't want to share the data. I think they have, like, good intentions, but they're not following through in the right way. I think that the more that we can all share examples where we have shared data, I think that's a good thing. Maybe some, like, social pressure that, like, these other groups and other people are doing it, I think it is a good thing. But I don't have a good answer because a lot of times people will hide behind their consent and say, well, the consent doesn't allow it. And sometimes the consent does not allow it. But oftentimes I think that they're using that as an excuse. But what do you guys think? Carla, do you have ideas? Paul, do you have ideas?
I was just going to say and throw this out there. I'm not sure it would work. Maybe people would hate it. But you know, we have groups like Charity Navigator that rate the efficiency of charities and in terms of their transparency and how much money they spend on administration and other things. Would it be possible, Dave and Carla, to create, through maybe, a nonprofit, ethical center, just a rating system that would be very objective about transparency, accountability, publishing frequency, it has the registry made available a data dictionary, just kind of a nuts and bolts efficiency score, and for how often that registry publishes and the quality of the peer-reviewed journals it publishes, and just make it as neutral as possible, and then just score it, and let the community and funders say, wow, okay, we got this score. That other registry did a little better than we did. How come? Why? I mean, I would just want it to make it easier for people, especially patients who are thinking about contributing their data to one registry versus another registry, should have the same ability to have an apples-to-apples comparison as much as possible.
I love it. I think that's a great idea, and may address some of the reasons why data are not shared. I mean, it's along the NIH. You submit an NIH application. I just did. Tireless nights. There's a data sharing plan in there, right? You have to write that in. How do you plan to share this data, especially if you're going to be funded? If these registries are funded, there should be an obligation of like, how will you share this information? Why things don't go share? I think the understanding the data and its provenance and what it means and what gets contributed is a real thing. Partially, some of it is protective, but there's a real thing there. But should be an opportunity to ask the question and collaborate. I think what the data holder might be concerned with is like, I can't pursue all the questions that come here, but I need to figure out, prioritize, how important is this? Is there a way to partnership to get funding to pursue this question so that we can work together on it?
Every registry should have a data nutrition label.
This is very
Exactly.
unhealthy registry.
Very healthy registry.
How healthy is the registry? Just go down and let us know.
We're doing a lot of this nutrition labeling for data, actually. This is a lot of the work we're doing
There's been a lot
now.
of discussion about labeling AI, like how do you label an algorithm and its performance. But I mean, I'm being partly facetious, but also 100% serious. Like, we just need a better idea of what good looks like, what responsible data looks like, and people should be making decisions about where they send their data and, based on the most responsible use and reuse of that data.
How do you protect that culture, though, to ensure objectivity and that the power dynamics don't play into the end goal?
Yeah, I think that's a great question. It probably needs to be housed in some neutral, probably an ethics program or bioethics program or something like that.
Great. Next question.
Thank you so much. My name is Nick Siever. I'm here really with two hats. One is I oversee investments for a family office investment firm where most of our work is in early stage biotech. And more importantly, I'm the father of a child with a monogenic disease called MED13L. First of all, this is fantastic. Thank you. Dr. Fajgenbaum, I wanted to ask a little bit more about the example you gave of being able to administer a drug just 2 weeks after the readout from I think it was the Somologic work that you did. I'm going to give a little advertisement for a company called Rarebase that our foundation is working with, trying to repurpose small molecules for our disease. And we're about to get readouts and really thinking about how to get these drugs to our children. So I'd love to hear a little bit more about how you were able to make that happen.
Sure. First off, 30% of all prescriptions written by doctors are for drugs that are not approved for that indication. So doctors are constantly writing prescriptions for off-label drug use. Most of the 30% of prescriptions are sort of established off-label, like there's a good body of knowledge behind using that drug for that disease, even if it's not approved by the FDA for that disease. There's a lot smaller percentage that's sort of the unestablished off-label. And so what you're describing is unestablished off-label, where basically we figured out from the proteomic profiling that there was this JAK1/2 signature. We went in the lab and we did some confirmation experiments, confirmed there's increased STAT3 reactivation. And then, like I said, I was having phone calls with this doctor in Chicago for about a year while this young patient was in the hospital, and she was just getting to the end of her road. And the question was, is there just anything that you're coming across? And it's, yeah, this JAK inhibitor, a drug called ruxolitinib, it's been around for about 5 years. It was developed for a form of cancer. And so I said, yeah, you could try ruxolitinib, but I mean, there's very little data. It hasn't been given to a human yet. This patient at the time was 15 years old. So like, you, the doctor, have to make this call. I can tell you scientifically, this looks promising. And in the oncology world, especially in the oncology inpatient world where someone's like dying in the hospital, there's a lot of freedom to try these sorts of things. And of course, a drug like ruxolitinib is actually a lot less expensive than even just spending another month in the hospital. So from an insurance company perspective, they're gonna potentially be in a position to repurpose or reimburse. For a hit that you get from your screen that is in a child that is maybe outpatient, and there's even less data, it's hard to then make that justification both for the doctor, but then also now the payer has to reimburse it. But the stronger your data, the more likely it is that both the doctor is gonna say, yeah, I think this makes sense, and the payer is gonna say, yeah, this makes sense. And I should make one last comment, and that's that you might find a hit, and if your doctor and if multiple doctors don't think it makes sense, there actually may be a really good reason for that. I'm not just saying like we should just repurpose everything for everything, because there are good reasons not to do that. It may throw off the risk-benefit profile, but that's how we did in our case.
Thank you.
Hi there, Chantel from Styrus Research. Hi, Paul. We had some great conversations last night, and I love hearing it being reflected up. David, this question is actually for you from a conversation that Paul and Craig and I had. So you talked about using Janssen's samples that they had collected from patients in studies and had sitting in the freezer for years. And this is exactly what we talked about last night. Can you talk with us about what it took for you to be able to get that data? And when we run the clinical trials and we say to patients, we're going to take some extra samples and we're going to store them for analysis later on, we don't necessarily say to them, we're going to sell this or give this to a university who might look at it and then take this and sell it to or give it to a bunch of different companies who might develop some things. Where do we sit with that, with sharing that with patients, and how do we avoid the Henrietta lack of, I took your cells and all of these companies have made a lot of money and you patients are struggling. So I'm just looking for some feedback on how you got around that, what you had to do, and any hurdles.
Well, the first answer is that it really does matter what was in the informed consent, because as I mentioned, a number of these patients are no longer alive, so we can't go back to them. So it is what's in the consent, and in the consent it did say that a number of different experiments could be done on their samples up until 10 years. And people, and I suspect Amicus is more broad, there's definitely a general movement to not put like a 10-year horizon on things. There's a general movement to be really free with that consent, to say many different things can be done by many partners, for-profit, non-profit. Generally, they, as things stand in 2022, they don't usually say you will receive any sort of financial benefit at the end. But yeah, what's in the consent is really important, and then how we actually made it happen was just spending a lot of time with our partners within the pharmaceutical company, and then also a lot of attorneys involved to make sure that the samples could be shared. And it's hard. It's really hard work. I don't know if either of you guys have anything you'd add to that.
Matthew Ellen with National NPS Society. This has been a great conversation. I really appreciate it. A couple of observations. The idea of silos of data, I kind of like to think of them as repositories. And as David mentioned, the consents really have an impact, and it's important for us to remind patient communities that in some cases, the people that curate that data may not have a choice, but they don't articulate that their hands are tied in some respects. And many of these date back to consents that were a best of practice 20 years ago, but now they're really looking much more forwardly and also empowering patients. So the Multi-Regional Clinical Trials Center at Brigham and Women's is a wonderful resource for these sorts of broad consents, and I think they should include also patient empowerment and return of data to patients, and even in the case of registries, the ability of patients to, or participants to withdraw their data so that privacy is really observed in every case. Thank you.
Good morning. My name is Ben Forrid. I'm from an academic background, and I lead a team of clinical researchers at a hospital system. But I also direct a rare disease registry called CORDS. And I just wanted to join the conversation on siloing of data and sharing of data in the context of a group that tries to run a disease-agnostic registry as best as we can without having any of those barriers present. I do think there's a need within this space for there to be multiple third-party, unrelated groups that are collating and collecting data and sharing that information out, and so that patients have the option of always owning their data, always having the toggles on where that data is going to go, and then opening up doors and opportunities for future researchers to freely access that data. And so I love the discussion that we've had so far about the consenting language and the barriers that advocacy groups run into when they host a registry in any one particular place. But I'm seeing a great amount of success that comes out of working with a third-party disease-agnostic group to host that data in an FDA, GDPR-compliant manner and really accelerate the pace of things. So just a little comment from my perspective.
Hi, really great discussion. This is Simi Ahmed. I'm with NICEF. We use the power of stem cells for disease modeling and drug screening in vitro to kind of de-risk the kind of failures that you've already discussed. So my comment really was along the lines of incentives. Everybody's talked about incentives and just sort of the source of where these newer or transformative incentives could come from. Could belong with funding agencies, as has already been mentioned, academic institutions for promotion and tenure, and even the publishers who can encourage collaborative publication. So I was wondering if you had any high-level comments on how those kinds of transformative changes could be brought about, or if there's something remiss that could be addressed.
Yeah, I mean, David and I worked on a commentary that I think we're still hoping to get out there, where we raised a number of those solutions, and Carla spoke about them too. I think the NIH is trying to do a much better job in terms of requiring data sharing and having a plan for data sharing. I think academic institutions can, when they're making decisions about hiring and tenure, really emphasize the creation of high-value data sets that get reused, that creating a super high value data set is incredibly valuable and can really advance the field. So I think there are lots of different ways that we can take responsibility for the pieces that we control and be more responsible for breaking down those silos. But I'd love to hear what my colleagues have to say.
Yeah, I would just add, and we'd spoken about this on our pre-call, that one of the biggest challenges, I think, is that a lot of academic research centers, they give tenure based on the number of first and last author publications. And so by definition, every paper can only have one first and one last. Sometimes you'll have a co-first and a co-last. So maybe there's 4 people that can be in those positions. And so when you think about your researchers that are part of your field around the country, if they all combine their data together, let's say 10 researchers combine their data together, and there's like an incredible insight that comes from these 10 different data sets coming together, there can still really only be 2 first and 2 last. And so by definition, 6 of those contributors are going to be quote unquote middle authors, which as a patient and for all of you, who cares whether you're a middle author or first author? But unfortunately within the academic research system, those 6 people are like maybe not going to be able to bring food to their kids' home, because they're not going to get promoted. And so instead, rather than 10 people combining data together, all 10 will publish their own papers and they'll all be first authors and they might get promoted and they'll be able to bring food home to their kids at night. And so I think it's really important as a patient that makes me sick to my stomach, but I think it's important for us as advocates to remember that that's the system we're in. Our researchers all around the United States and academic centers, they're getting graded by the number of first and last author publications. There's only so many positions for that. So I think we really have to, I don't know how to do it, and I hate in these firesides to propose something that I don't know how to fix, but we got to figure out how to fix that big problem.
So to this, in networks, so again, the Innovation Medical Evidence Development Surveillance Network includes 9 different healthcare delivery and healthcare systems. when it's the same problem, right? We've got all these researchers together, and then we're doing this study, and then who gets to do what? So the model is what it is, but what we try to do is early on say, these are the things that we think are going to come out of it, and let's divvy it up. Like, who's going to take first on this? We can get 4 papers, 4 different first authors. Divvy it up. I mean, and somebody has to get something because they need to feed their kids at night, and they need to pay their research staff, more importantly, because you can't hire people and then not have funding later on to keep them. So it's a reality.
Thank you.
That last answer was a good segue to what I wanted to ask. I'm a big believer in scholarship and transparency, by which I mean peer-reviewed publications. And I'd like to get your take on whether there is value in journals that commit to publishing negative results, especially in the rare disease space. Do we have any journals out there that commit to a certain percentage of their publications addressing this? And the second question is, PIs, especially in academic environments, never want to admit they can't get anything done. At least that was my experience. I certainly hated doing it. Is there value in just advocacy organizations funding medical writers to get this stuff out? Because I know a lot of registries, the only mechanism to disclose data is a peer-reviewed publication. So thank you to discuss those a little bit.
Well, I don't know about, is the question about to commit publications to commit to publishing negative results?
Yes,
Or
especially,
failed trials?
especially regarding rare diseases.
Right. I know that BMC has done an extensive review of failed protocols, and their commitment is that when you're publishing, that you publish results from all the protocols, because there's so many, failed trials that have not been published. And it's right to this question is like, they've issued a big commitment, and I think Peter Doshi was one of the main editor who published an article on that, and that journal is specifically focused. That's one of them. I don't know about others, but
eLife is a journal. They will publish replication studies, and whether or not they're positive or negative. So there are a few examples already that have been published. The other question about medical writers, I think that is definitely a good solution to getting the publications, I would say, out the door. I met a medical writer last night. I think this is an area of growing interest for communities to find people who could write not just in the vein of popular press, but actually within nuanced professions, technical writing. So I think we are out of time. Let's thank our panelists. Give a round of applause.